So tell us a little bit about your study and the findings.
Great. So hematopoietic stem cell transplant has long been a standard of care for treatment of inherited metabolic disorders, both to halt the progression of these diseases and also to combat any neurological complications. Cord blood transplant is a standard of care for select inherited metabolic diseases where there are neurological complications. But cord blood typically has low numbers of hematopoietic stem cells, which ultimately leads to delayed engraftment and graft failure in about a quarter of patients. At Magenta, our goal is to develop therapies where we could have high numbers of high-quality stem cells. To this end, we developed a cell therapy called Magenta 456, which contains greater than 300-fold higher numbers of stem cells relative to standard of care. If we take Magenta 456 and transplant these into mice, we get higher levels of engraftment in the periphery, but interestingly, when we look in the brains, we also see higher numbers of a hematopoietic cell type called microglia. And this effect is both rapid and sustained. We see higher numbers as early as 2 weeks post-transplant. So why is this important? Because if microglia are the cell type which, after transplant, can engraft in the brain and lead to cross-correction of neurological defects. So our data suggests that Magenta 456 leads to higher numbers of hematopoietic stem cells and other cell types that are important for disease cross-correction.
Great, and you've done this in a mouse model?
Absolutely, yes.
Great, okay. So what are you guys thinking in terms of which disease states this potentially could be applicable to, recognizing that, obviously you're in early phases of research at this point?
So for the diseases, this would be subsets where there are neurological symptoms, like cerebroadrenoleukodystrophy, Hurler syndrome, etc., because these are the diseases that we want to be able to get higher numbers of microglia into the brains of patients for cross-correction of these neurological symptoms.
Got it. And so what are your next steps? What are you guys going to do going forward now that you've sort of discovered how this works in the mouse model? Are you planning any further animal studies or potentially early human studies?
Yes, so there's an ongoing phase 2 trial now led by the University of Minnesota in select inherited metabolic disorders. So we'll be interested to look at disease outcomes in those patients.
Great. So we look forward to hearing about the results maybe in a year or two?
We're actually presenting some updated results at AAN this year,
Okay.
Tomorrow evening session.
Great.